Hello, everybody, and welcome to the newest podcast of the Canadian Open Neuroscience Platform, CONP. In today's episode, we will be talking with Alexander Jacob, PhD student in psychology at the University of Toronto. With CONP's support, Alex is currently working to develop new open source tools for imaging brain activity in awake, behaving animals. Let's hear what he has to say. So welcome to the CONP podcast. It's very good to see you and meet you.
Yeah, thank you so much for having me.
Can you maybe tell us a little bit about your career path? How did you come to do research in neuroscience, and what led you to apply for the CONP Scholar Program?
Yeah, sure. So, I got my start in neuroscience kind of in a sort of backwards way. I was always interested in science, going through school in high school. I was also really interested in theater and doing backstage theater stuff. So there came a point kind of when I was applying for universities where I had to decide, which thing I wanted to do. And I figured that you can do amateur theater and professional neuroscience. That's okay. But if you do professional theater and amateur neuroscience, you can get into some trouble. So I picked neuroscience, and I've been, I've fallen in love with it ever since I first started. I've been really lucky that it's been a good fit. So and sort of as I went along, I've kind of pursued more of an engineering bent, though I'm in a psychology department. I think the really cool thing about neuroscience is kind of how many different fields you can reach out and touch if you just look in the right areas. So it's been a really, really interesting process, and obviously I'm still in the middle of it. I'm not even done my PhD yet.
And where are you at the moment? You're at the University of Toronto, correct?
That's correct. Yeah. So I'm in the Department of Psychology at the University of Toronto, and my lab is affiliated with Sick Kids, the Hospital for Sick Children. So that's where we do most of our work, is down here at Sick Kids.
Are you Canadian by birth?
I am. Yeah. I was born in Edmonton.
Right.
Great city, Paris of the West. Yeah, very nice. But I came to Toronto when I started my undergrad, and I've been here ever since. It's a great city to work in.
I assume quite a bit warmer too, huh?
Yeah. The winters are more tolerable here than they are back out west.
Now, I noticed that you are active on Twitter. You're NeuroChatter on Twitter?
Yeah, that's right. Yeah, that's right.
How long have you been tweeting?
Just a couple of years. I have to say I'm not too terribly good at it. Some of our other lab members and Sheena, my supervisor, are a lot better at it than I am. But it's fun to connect with the science community that way. Twitter's a huge resource that I didn't even realize existed sort of at the beginning of my grad school career.
And you said you're also an audio editor for a podcast?
Yeah, that's right.
Care to tell us a bit about that?
Yeah, sure. So the podcast is called Raw Talk. It's done through the Institute of Medical Science here at the University of Toronto, and every 2 weeks we have a new interview with some Toronto academics, usually working in the medical field, and also just sort of touching on topics that we think are interesting to the broader public about science. So it's been a really cool project. I just started on it, but they've been doing good work for, I think this is the 4th season. So you can check that out if you're interested. Exciting. But yeah, it's a fun way to sort of extend our findings beyond just the sort of like talks and immediate research community.
Well, you know that we could always use more contributors for the CONP podcast, and honestly, sometimes we're looking for ways to cross-pollinate and share content across platforms. So
sure.
If you'd ever like to contribute, either by doing a CONP podcast, or maybe just letting us know that you've done podcast that might be of interest for the CONP, it's nice to just grow the community.
Yeah, absolutely. That would be great.
This is good to know. So
yeah, absolutely.
We've already made something hopefully useful for both of us, for the CONP and for you, by syndicating some content.
Exactly. I think that would be really interesting. And the Canadian community is really cool. The Canadian open science community has been really great. I think because we're a smaller country, it's easier to make these connections and cross pollinate, like you said.
Yeah, as Alan Evans says, usually the right country is big enough to make an impact, but small enough to be manageable.
Exactly.
So I think Canada qualifies.
Yeah, no, that it's been really awesome working in Canada.
So, can you tell us a bit about your project? What are you working on at the moment, and what was your CONP Scholar proposal for?
Sure. So the project that CONP is supporting me on is work we're doing to develop open source miniature fluorescence microscopes. So there's been some really exciting advances in the field. My lab, sort of traditionally looked at behavior and how what are the neural correlates of behavior. And I guess about 7 or 8 years ago now, some folks out of California developed a new technology where they're basically able to miniaturize a standard fluorescence microscope and make it so small that they could put it on the head of a mouse. So you could implant a lens into the mouse
So,
brain.
how small is that?
It's just a couple grams. It's maybe 10 centimeters tall. The mice just wear it on their heads and they can walk around and interact with each other and do all sorts of behavior. So we're talking right down kind of at the the limit of sort of standard sort of commercially available tools. So we use
Very impressive.
we
And
use
how about the surface area of the microscope? Like how big is it on top of the head?
Sure. So I guess it's about now. I should look at the measurements again. I work with these things all the time, but honestly, mine for these things isn't so good. It's about maybe I don't know, 2 centimeters by 2 centimeters, maybe a little bit larger than that. Probably more like 4 by 4 is the
That's very
big
impressive.
footprint. But the lens itself that we image through is 2 millimeters in diameter. So
So there's
everything's
these mice walking around with kind of a cylinder on top of their head.
exactly, yeah. So it's really interesting. I mean, it's made up of basically just commercially available parts. Actually, a lot of the things that we use are the same technology that's in your cell phone. We have a little LED that provides the light that goes down into the brain. That's just the same as you would see on a cell phone or something like that. And the camera is actually similarly the big advance. That has helped us a lot is these miniaturized CMOS cameras, and those are the same things that you can use to take nice pictures on your phone.
Cool. And you're recording in real time? You're recording all the time, or
Exactly.
is it
So
only when you're
we
performing the
we
experiments?
typically only record for the duration of an experiment, which might be between 10 and 20 minutes, perhaps longer. There are biological considerations to take into account when you're looking at the bleaching. So we combine this technology with a genetically encoded calcium indicator. So what that means is that if you're imaging all the time, you're eventually going to degrade the proteins that are being made by the brain. So our recording sessions are short, but it's long enough, and it is in real time in the sense that we can correlate what the mouse is doing at a particular time point with activity in a population that's maybe several hundreds of neurons large.
Cool. And how do you stabilize the image? I imagine that mouse motion is not very easy to correct for.
Yeah, no. So this actually touches, I think, on one of the most interesting components of this project, because you're right. The images that we get out aren't perfect right out of the box, and so there's actually a lot of work that's done on the computational side by some of my colleagues in the lab and other CONP supported scholars, looking at how we can not only stabilize the images but then actually extract information out of the images, because you have to imagine what we get out of these microscopes is just a video of a bunch of blobs, getting bright and then getting dim again. And from that, we have to extract information about what are the cells, what is their activity over time, how are they correlated, and then from there you can go on and do analyses to look at sort of how the network is behaving. But you're absolutely right that there's a big computational aspect to it. The motion itself can actually be corrected for a little easier just by engineering things properly. So if your connection to the mouse head is stable enough, you don't actually get that much motion. But there are absolutely things related to that we need computational support to get around.
Cool. So, what is your open science deliverable for this project? What aspect of this project would you like to make more transparent, more reproducible, maybe?
So we're interested in kind of every step of the process. Like I said, there's definitely an engineering component. That's the actual physical construction of the microscope. How it works, where to get the parts, how to assemble it, and that's the thing that we're focusing on most closely now. Both making our current design more open, and of course we're working on new designs to deliver things that weren't available in previous versions of the microscope. And continuing to develop those and make those open. A big focus of our particular project, because we're not the only ones doing this. There are actually a number of other great groups out of the States. Miniscope, originally out of UCLA, is one of the biggest, and there are several others. There's one that they use in birds. I think it's called FinchScope. But in general, all of these projects are providing their resources online in the open, and we've done the same mostly over GitHub. So continuing to communicate both how to construct these scopes and the best ways to use them, so that labs that maybe don't have that much of an engineering background, and that's certainly the case for us. We didn't start as an engineering lab going into this. So making the plans and construction available for the wider community. And then as a second part, in addition to the engineering, there's also the computational side. So that's making code available to better analyze these calcium imaging videos and better extract cell signals that you can use for your particular analyses. So those are the main deliverables from this project.
Cool. So when you make this microscope open source, do you expect it was going to be relatively cheap to make one, to build one?
Yeah, so that's been the one of the real focuses of our design, is making sure that the cost of parts is achievable for labs of any size. So right now, the design that we're using costs about $1,300 to make, 1,300 Canadian dollars. The majority of that is the camera, but overall, that's pretty affordable for a single system. By comparison, there are a couple of commercial companies that are making these available for sale, and one of their packages is about $150,000 for one or maybe two scopes.
So two orders of magnitude.
Two orders of magnitude cheaper. So that's been the real advantage, and sort of the real reason why open science has been the winner here, I think, is because we're able to make this technology available to such a much larger group of labs who can use it to answer questions that they're interested in, instead of keeping it at this level where only big research groups are going to be able to afford it.
Cool. So, it's very impressive that you're trying to make very expensive technology widely available. Would you say that this is a philosophy that your entire lab has? Is this something that is the modus operandi of your lab, or is your project kind of an outlier in terms of transparency and openness?
I think the general focus of transparency is one that our lab has had for a while. It's actually been really interesting. When you look at a project like this that involves a lot of engineering and develop, sort of building of new systems, that's where it can be really easy to make a project open, like we were talking about, right? You can find a way to make it cheap, and then you can make that available. A lot of the things that we've been doing up to now have mostly used, those commercial systems I was talking about earlier, not for imaging, but for the sort of behavior we do. And making those open has been a lot more challenging, because I think labs in a way are small c conservative. They want to pick something that works, right? So sometimes that can mean buying from a supplier. But as we move into spaces where we are more able to do things ourselves, the focus really has been on sharing that with the rest of the community, because we all benefit when we are using the best techniques and technology. So
what are your career plans? What do you hope to do after this project is finished?
That's a great question. Obviously I'd like to continue the sort of work that I'm doing here, which is, pursuing curiosity and answering questions and developing new techniques that hopefully, allow people to answer their own questions in interesting ways. So as for how I will realize that, in my particular case, it's not immediately clear. Obviously, the postdoc is one way to do things. But even in industry, we're seeing more and more kind of interesting positions for neuroscientists opening up. But it's sort of melding that, potential for working in industry with hopefully, continued focus on being open. I mean, I think that there are a number of open source software projects that are really interesting. So it's a matter of seeing how those move into the neurospace, on the industry side, that I'm still interested in looking at.
So I guess thinking one level higher, just about the way that science gets practiced these days, were you to continue with an academic career, which is something you're definitely considering at this stage, is there any aspect of science that you find slow, unproductive? Is there one aspect of science that you would like to see done differently?
Yeah, I mean, that's a big question. I think that science naturally really wants to be open, and I think that there have been a lot of strides made recently, to make science a lot more open and to share techniques a lot more widely. I think that for all the communicating that scientists do, sometimes scientists are very bad communicators, especially when it comes to things like developing a technique and then maintaining it. I think that in computer science or software engineering has done a much better job of putting an emphasis on making things reproducible, of developing a product or a technique that works not just for you but for everybody. I think in science, because we're on the cutting edge, so often we will build something and then it works, and then we share the plans, and then we say, okay, we don't have time to support this, we're a small group, we're going to move on. So one of the things that I'd like to see is in science is some way of balancing the need to continue to innovate with the need to support what you've already made and communicate it effectively, both to the public and even to other research groups. Because otherwise what you wind up with, and what I've seen a lot of, is there are a few, large research groups that have the expertise, that have the techniques, that are able to do things. But their results don't necessarily, it's not that they don't replicate, it's that other people don't have access to the techniques to even check their science and make sure it's robust. So that's, I think if I could make one big change in science, it would be a continued emphasis on really incentivizing a way to share our ideas more effectively and continue to maintain that, because right now there's not so much of an incentive to do that necessarily.
Cool. Well, you know that we do have a communications committee as part of the CONP. So if you ever want to join our meetings or contribute content, we would love to host it.
No, actually, the CONP has been super exciting because it's like I was saying, if we need institutional support to try to bolster our ability to do open science, I think that CONP has been really helpful in that, in providing actual monetary support, putting money on the line to say, yes, we want open science in Canada, and it's an important goal.
So outside of the lab, who is Alex Jacob? You mentioned you do theater. Do you still do it?
Not so much anymore. As I've kind of progressed through the PhD, things have been gotten busier and busier, which is one of the reasons that I kind of wanted to get back into doing something else, and so I started working on Raw Talk. Was I always really enjoyed the theater work that I did. I think it's really exciting to see a show come together. I think in a lot of ways it's like seeing a paper come together at the end, right? Things look messy and they don't always work, and then right at the end you can usually pull something out that is a pretty satisfying end product. So that's what I tend to do, other than relax, watch Netflix, play video games, go on walks. Yeah.
Cool. Give us a Netflix recommendation.
Oh man. Oh, that's so tough. I actually only recently got into Fargo, which is a TV show set in, it's sort of about crime in the Midwest. And
have you seen the movie, the original Coen Brothers
movie? I saw the original movie, and so I thought the TV show would be very similar, and it is in some ways, but it really isn't in others. So that's been a wild ride. I think my favorite show that I've watched in the past year is probably Maniac, which is a Netflix original series. That is a show that is
with Jonah Hill and
Emma
Stone.
Emma
right?
Stone.
I saw it.
Great show.
Cool. Well, seems like we have similar tastes. So hopefully that's an opportunity to catch up again and chat more.
For sure.
I wish you good luck with your program, with your PhD. And do get in touch when you have something exciting to share. We would love to do more to make your work visible.
Absolutely. Thank you so much, Nikola.